Thanks, Tridib, Kavita, and Sakuntala for this invitation. It's an honor to be part of this wonderful occasion. And let me start by thanking Mustansir and Deepak for the guiding, motivating, and inspiring generations of physicists in statistical mechanics in India and all over the world. Here, I just wanted to highlight the youthful exuberance that remained constant over time. It's a privilege to note two of the finest theoretical physicists and human beings. Belated very happy 70th birthdays, and wish you many happy returns of the day. So I'll be talking about cell shape. Cell shapes are important for different cellular functions. Different biological functions are actually interplay of very complex chemical signaling networks. But there are enough evidence that cell shape and these chemical signaling networks, they are coupled. So this mechanistic point of view is basically that cell shape and this interaction network, this coupling. So the mechanistic point of view is that one can ignore this coupling or treat it as a black box and study the function in terms of cell shape. So cell shapes are important. Let me just The cell shapes are important for both in health and diseases. For example, in human beings, there are about 200 different types of cells with varying shapes and different functions. Cell shape changes during different processes, such as during cancer progression, asthma, wound healing, development, etc. And cell shape affects crucial biological functions as well, such as the growth and apoptosis, division, plane orientation, differentiation, and morphogenesis. Moreover, cells are the functional units of tissues. So tissue shape must originate at the cell shape. But how it happens remains one of the fascinating questions of developmental biology. Till now, this relation between shape and function are mostly studied via average cell shape. But as you see in these figures, that cell-to-cell shape also varies. So how about the cell-to-cell shape variabilities? In 2018, in this fascinating paper, Atia et al. Showed that the cell-to-cell shape variability across diverse epithelial monolayers follows a nearly universal distribution. So they characterize the cell shape via this aspect ratio, which is the ratio of the long axis to the short axis. And they measured, so these are the measurement of the aspect PDF of the aspect ratio. And when they plot this particular quantity, the ratio of the aspect ratio to the average aspect ratio, they find that the probability distribution function of that quantity X follows a nearly universal distribution. So they fitted this this data with the K gamma function, which is noted here. So this has a single parameter K. And when they fit this function with the data, they find that for diverse epithelial systems, K is nearly the same. It is around 2.5. Although there is a slight spread around this value. Moreover, they showed that the standard deviation versus the average aspect ratio follows a universal relation for different systems. But several questions remain. First of all, what is the origin of this universality? What sets the specific value of K around 2.5? What are the consequences of these universal behavior? And these are some of the questions that we addressed in this work. So this work is a theoretical work. In the theoretical approaches to understand the static and dynamic properties of cell monolayer, the cells are represented as polygons with a preferred area and a preferred cell perimeter. One crucial aspect of this system is that the constant of confluency, that is, there is no gap between cells. So the total cell area is the total system area as well. So there are different models, as I was talking about, that there are some continuum model and there are some discrete lattice-based models. The energy functions of these models are basically this following, this H which is given as this. AI and PI are individual cell areas, and A0 and P0 are the target cell area and the target cell perimeter. N is the total number of cells in the system. Lambda A and lambda P are two area and perimeter moduli. So they basically characterize how strongly these two constants are satisfied in the system. So given this energy function, one can study the dynamics in this system via a temperature T. this temperature in this system are some type of effective temperature. So they characterize all types of activity, including the equilibrium temperature. But within these models, this temperature has the status of an equilibrium temperature. So given this energy function and within this model, our goal is to study or calculate the distribution of the aspect ratio in a monolayer, in a confluent monolayer. These models are quite high level of simplification of the actual system, but even with this simplification, a direct calculation is actually challenging. So we took a slightly different route. We first calculated the distribution for a related quantity, which is the radius of gyration around the center of mass, and then obtain the desired distribution of the aspect ratio. So this is written this probability distribution of the squared radius of gyration is squared. Z is here the partition function. The first delta function basically coincides the center of mass of the system with the origin of the coordinate system. The second delta function picks out specific values of the S squared. And this exponential is like a Boltzmann factor with the energy function given as the energy function of the system. And this last part is the volume element. So now this expression is quite general, but from here to get a useful expression is quite non-trivial. And we had to make several simplifying assumptions for analytical tractability, and then we obtain this particular expression. So P of R is given as this, where N is a normalization coefficient. We have actually exact expression for this, but this has this particular form. Now, the point to note here that all the system-specific properties combine into a single parameter, alpha, and cell shape variability in the system is described by the single parameter. And this has important consequences for the universal properties. So first, let me explain some aspects of the universality. In this expression, this factor 3/2, it comes from a geometric property. And the geometric property, which is the cell perimeter, is a closed loop object. So this must be generally true for all systems. And these are some of the PDFs of the aspect ratio at different values of alpha. Now, as they did in the experiment, we also calculated the distribution of the scaled aspect ratio that we denoted as RS. And when you plot the PDF of the scaled aspect ratio, they follow nearly a universal behavior. So they are very close to each other, although they are not strictly universal. There's a slight variation in them. The second point that when they in the experiment, they fitted the K gamma function with the data. So if we take this particular approximation that ignore 1 over R with respect to R, then from here we get the PDF of the RS, the scaled aspect ratio, as the K gamma function. But this approximation is not very good as R is of the order of 1. And that basically leads to this slight variation in K when you fit the K gamma function with the experimental data. And this value K of the order of 2.5, so it comes from this geometric property. It's a consequence that this particular factor is 3/2. So it comes from this geometric property that cell perimeter is a closed loop object. This next aspect is that this distribution function is given by a single parameter alpha. So both the standard deviation and the average aspect ratio is determined by the same parameter alpha. So we can write down the analytical expression. The standard deviation is a function of alpha, and average aspect ratio is another function of alpha. So from these two equations, we can write a parametric equation with the SD given as a function of the average aspect ratio. Now, these equations are monotonic, and we see that alpha is does not appear in this equation. So what it implies that this relation must be universal across different systems. And this is the theoretical plot of this particular quantity. The actual functional form is very complex, but it looks nearly linear. So then we tested all these predictions as well as some of our hypotheses in the simulation. And let me briefly just point out that it is consistent. So one of the predictions that the PDF of the scaled aspect ratio is nearly universal, which is seen in our simulation as well. For vertex model and CPM, we are showing this data. And this relation between standard deviation with respect to average aspect ratio. So this is this dotted line is the theoretical plot, and these data are the simulation data. And they also agree with our prediction that this must follow the same relation. Now, then we compared our theory with experimental data. So we took experimental data for this particular figures from this paper by Atiyah et al. In Nature Physics. We took the PDF of the aspect ratio, and these lines are the theoretical plots and symbols are the experimental data. And we obtained the value of alpha. Once we have alpha, we also obtained the PDF of the scaled aspect ratio, and we then compared with the experimental data. Again, the agreement looks very nice with the experimental data. So the point is that the theory is consistent with the experimental data and the interpretations that they had in the paper. So now one of these very strong prediction of the theory is this PDF of the scaled aspect ratio is nearly universal. So what we did, we took data from different papers for a variety of systems, and then we plotted them on the same graph where these lines are our theoretical predictions for two different values of alpha. And this nearly universality aspect that we predict from the theory also comes out from these experimental data. So the variety of cell types in this plot is actually quite remarkable. There's various cancerous cells, there are various asthmatic, non-asthmatic monolayers, yet this nearly universal behavior is consistent with our theory. And these are the references that from where we took this data. The other aspect is the nearly universal behavior of, the universal behavior of the standard deviation with respect to average aspect ratio. So this is a consequence of a single parameter in the distribution. And so we also tested it in experiment where we took data for different systems from different papers that we have listed here. And again, the agreement is quite remarkable. Here, this theoretical line is this solid line, and these are the experimental data. So this is not a fit, it's just the plot of the theory and the experimental data. Now, just at the end, let me just point out this predictive power of the theory. So there is one parameter in the theoretical distribution. So we can have an analytical expression for the average aspect ratio with alpha. And once we know the average aspect ratio, we can calculate alpha from using this relation. Once alpha is known, all properties about the distribution is known. So we tested this this aspect in experimental data. So we collected data for the average aspect ratio for 6 different systems from this paper, and then we obtained alpha from here, and then plotted this PDF of the aspect ratio. So we didn't have data in the paper for this particular quantity, but we had data for the scaled aspect ratio. And these are the plot of the theoretical plot, and then these are the experimental data. So to conclude, distribution of the scaled aspect ratio is nearly but not exactly universal. The standard deviation versus the average aspect ratio is universal. And the source of this value of k equals to 2.5 when experimental data is fitted with k gamma function is a geometric property. That is, the cell shape, cell perimeter must be a closed loop object. Thank you.
Thank you, Professor Nandi, so for such a great talk. So we have a question from Sanjeev Sabha Pandit. So you can ask. Professor Sanjeev Sabha Pandit.
Sanjeev, you are muted.
I cannot unmute. Okay. Yeah, yes,
now.
yes, sir, you were.
Yeah, I was not allowed to unmute. So, yes, so see this, it's a nice talk, and I also quite intrigued with this, that, okay, you can fit a lot of this thing by a single curve. But if you go to your this probability distribution function, see, of course, there is an essential singularity near r equal to 0, but apart from that, it seems to be negative, right? Because of this minus 1 by r square. Isn't there a problem with that distribution?
Well, so r is defined as the, is this quantity that the long axis divided by the short axis. So the minimum value is 1. And it starts from 1 and it can become infinity. So then that problem does not arise here, because r starts from 1 and then it goes to infinity.
Okay, yeah. Okay, thank you.
Yes, there is one more question.
Yeah.
Sorry.
Hi, Sriram. Yeah.
Hi. I didn't understand exactly what you mean when you say it's almost but not completely universal. What that means is the dependence on alpha is so weak that it's practically the same, right? I mean,
exactly. That when we plot this function is very small.
So it does depend on alpha, but it's very robust to it. Actually, another little question, which is, so in this model, you start with this energy function. Does your I missed some details. Cell division and cell depth and so forth are not in your theory, or they are there? I didn't understand.
Yeah, so the cell division and the apoptosis are not there, but we have tested in the simulation. So here there can be two parameters. One is 3/2 and alpha. 3/2 comes from this property that cell perimeter is a closed group. So that cannot change. So it can only enter via alpha. So we did simulate with division and apoptosis, and in that case also this particular form works quite well.
I see.
So it changes the value of alpha.
Nice. Okay, thanks. Very interesting.
There is one question in chat box from Deepak Dhar. The picture of cells are two-dimensional, but the cells are three-dimensional. Can you comment?
Okay, so in a epithelial monolayer, the height of the cell remains almost constant. So this is an experimental observation. If the height remains constant, then one can write down a two-dimensional theory for that system. So all these theories are from that motivation, that the height is remaining constant. So therefore, one can describe the system as a two-dimensional system. But if the height fluctuates quite strongly, then these models will not work. One requires some type of modification there.
Okay, since there are no other questions, so let's thank Professor Saroj Nandi for such a great talk. Thank you, sir.